So the next one, I just want to share with you that the BRCA1, we already shown that as a co-activator for PBD3. That was done in the luciferase reportage assay. And this is just to show you again that the BRCA1, in addition to function as a co-activator of PBD3, it also a positive regulator of 14-3-3 sigma. Because you will see here, sigma itself is a PBD3 target gene. So you expect that will happen. And indeed, that is the case, okay? SCT116, MCF7 cell, if you transfect or they're using the adenovirus, carry the BRCA1 gene for dual infection. BRCA1 express will induce the sigma expression based on this Northern Blot study. Okay? BRCA1 induce the 43 sigma expression. Okay? And if you take a look at BRCA1 MEF, anybody know what MEF stand for? MEF, what's MEF stand for? You'll get bonus point if you know what is MEF stand for. Good, yeah. This answer, mouse embryonic fibroblasts. So these are basically based on the knockout mouse model, or we will be able to get the fibroblasts from that particular knockout mice, that particular knockout mouse, to get the mouse embryonic fibroblasts, stand for MDF. Okay, so that's a very important research tool. So you just compare the BRCA1 non-MEF with the wild-type MEF, you already immediately know that wild-type have more 14-3 sigma than the normal one. Okay? So now you know when the person do not have BRCA1 or have BRCA1 mutation, 14-3 sigma will be low as well. Right? Okay? And then we know 14-3 sigma do a lot of things. Okay? Here again, the sigma promoter just trying to find out whether BRCA1 really transcription activated 14-3 through the promoter regulation. Okay, PBD3 and BRCA1 there, then you see the 14-3 sigma promoter activity is high. Okay, if you add BRCA1 delta C, this is construct, will not be able to transcription activate the 14-3 sigma. You see the sigma activity got compromised. We talk about ATM, we talk about COP1. Okay, right now I want to show you another important regulation of ATM. Okay, ATM, if you remember, has many substrates. Okay, check 2, check 1, ATM, you name it. But the list could be longer, okay? Indeed, that is the case. ATM has another substrate, which is COP1, okay? You can see here, okay? This is the cell line being treated with ionizing radiation to cause the DNA damage, okay? And as you can see, following the time course of DNA damage, okay? PBD3 is elevated, okay, as expected. DNA damage causes PBD3 elevation, okay? You see that PBD3 is up, okay? The phosphorylation on the PBD3 is up as well. Interesting enough, the COP1, this E3 ligase is going down, okay? You see that following the time course is going down, okay? But if you're using an ATM minus cell to do the similar experiment, treated them with ionizing radiation, following the time course, the COP1 seems to be quite stable compared with the wild type, okay? So indicating that somehow ATM was able to, after the DNA damage, was able to manage the CAP1 downregulation. Okay. In the CAP1 downregulation, it's very important for its activity during the DNA damage response. I do not have the time to go further detail, but the bottom line is that ATM could cause a phosphorylation on CAP1. It's phosphorated on serine 387, okay? And then you can see that this ionizing radiation is phosphorylation dependent, because if we mutate that particular ATM phosphorylation site on CAP1, CAP1 is no longer downregulated, as you can see here, okay? You see the normal situation, CAP1 is downregulated after ionizing radiation, and compared with the no ionizing radiation, the CAP1 is downregulated. But if you mutate that particular site of ATM, CAP1 is stable, not affected by DNA damage at all. Okay? So the idea is ATM activated phosphorate on CAP1, CAP1 downregulated. Because the CAP1 downregulation, it cannot degrade PBD3. So ATM in the second pathway or in the third pathway will be able to preserve the PBD3 activity. Then the PBD3 to do the DNA damage checkpoint, to cause the cell cycle arrest, to do the DNA repair. So ATM goes through each pathway. ATM has direct impact on PBD3. We talk about its impact on serine 15, okay, causing the PBD3 stabilization due to this phosphorylation prevent MDM2 from binding to PBD3. Okay. ATM itself has an impact on MDM2 itself by phosphorylating MDM2 at certain sites, leading to MDM2 is no longer be able to bind to PBD3 to cause protein degradation. You could look up the paper. Sometimes I just want to share these knowledge with you, but in detail, okay, experimental. I know many of you still have no research experience here, but this is just at the beginning to let you appreciate the Western blot study, the immunoblot study, and the others to show how scientists try to characterize all these cancer cell signaling pathways to understand the regulation process. So we've been talking about DNA double-strand break, the activation of ATM due to the conformation change of the DNA double-strand break. The dimer of the ATM will be activated, and then they have a cell phosphorylation, and this cell phosphorylation in turn mediate a lot of substrate phosphorylation, including MDM2, we talked about MDM2, PBD3, CHEK2, CHEK1, BRCA1, MBS1, CLO, NCAP1, and etc., etc. You remember that on the slide I gave it to you. So many particular subjects. Is this list of substrate complete? I don't think so. Okay. So there must be other substrate meant to be characterized. Remember, we have such a small universe in our cell, okay? And then for them to run into each other, any protein to run to each other is very efficient, okay? So they will be able to regulate a certain protein. So right now, as a scientist, we're trying to dissect certain moment to see their interaction. Do they have the capability to regulate these proteins? In other contexts, maybe not in the DNA damage. What do they see each other? We don't know yet. Okay? Yeah, some people ask about stem cell. How about stem cell? ATM regulate these? We don't know. Okay? So if you have a stem cell research interest, you could always ask these similar questions. Okay. And then these are downstream regulation about P21 we mentioned a lot. It was able to regulate cyclin ECDK2, 1433 sigma, CDC25, a phosphatase to remove the inhibitory phosphate on CDK, okay, causing the G1 arrest. This is supposedly CDK1, okay. Even the scientists will make a mistake, okay. And cyclin B1 complex opposed to CDC1, CDC2 or CDK1. BSL1, MBS1, we talk about those, okay. The MBS1 and the mRNA complex, MIE11, REF50 involving DNA repair, okay. The stress response through the stress activated protein kinase. So it just give you some idea about all these DNA damage checkpoints, okay. I think we'll stop here, take a break, and then next round we'll talk about some of the growth factor regulatory pathway involving mTOR signaling, okay. Thank you.